Hello? Yeah, perfect, thanks. Thank you for coming. We are very privileged to have Professors Peter Tonelato and Dennis Wall from Harvard and Stanford today here. They'll be talking about how they're using Amazon Web Services to do whole genome sequencing in the context of cancer care in the clinical context, right? So before they come up on stage, what I wanted to give you was a brief overview of what Amazon has been doing in bioinformatics, why a lot of bioinformaticians are moving to Amazon, new platforms to do genomics analysis are being built on Amazon Web Services, and then we'll hand it over to professors who will go through what they've been doing at Harvard and Stanford. So we've been collaborating with the life sciences industry, not just in the genomics context, but a variety of other workloads from big pharmaceutical companies to a lot of exciting new startups that are developing platforms on top of Amazon Web Services. And that has been going on for quite some time, something that not a lot of people know, but the life sciences industry is embracing the cloud, has been using the cloud for quite some time. And we're excited to be working very closely with them. There's a lot of collaboration going on where they provide us a lot of feedback as to how to improve our services, what is it that they're missing that will benefit them, and then we work with the product teams to introduce new features or services to satisfy those requirements. One such program that we have is an AWS Public Datasets program. A lot of people over here may already know about that. It hosts the 1,000 Genomes dataset, and very recently we also announced the availability of the Human Microbiome dataset as well. Very useful resource, lots of data available on Amazon for free for people to be able to use and to do research. With Human Microbiome Project, we're also going to make available tools to be able to look at the data, analyze the data, and such. One more example of collaboration and work that we've done with another partner of ours, DNNXS, working with Baylor, has been with the CHARGE project, where they recently did analysis of around 14,000 individuals and looked at how whole genome sequencing and analysis and exome analysis can help in understanding aging and heart disease, and resulting in 400 terabytes of data that's available to researchers around the world to do follow-on analysis on. Just to give you an example of the scale of which the analysis is being done on Amazon Web Services, why people are moving to the cloud, because the infinite amount of storage capabilities and compute that is required by the researchers and scientists, and how cloud can give that storage and compute capabilities to the researchers. So to give you a sense of what are the building blocks that Amazon offers, so we have a compute platform, elastic, scalable, and what bioinformaticians love about this is it's a programmable resource that you can launch at the end of an API and launch from 1 to thousands in a matter of minutes. And it's a very flexible resource. You use it as you want it, optimized for your applications. We offer a variety of options for you, from high memory instance types to memory optimized instance types to compute optimized instance types. So a lot of capabilities to be able to do a lot of heavy compute analysis on your data. Now, where do you store the data? A lot of you are familiar with S3 as a scalable object store where you can store a lot of data, a lot of genomic data is going into S3, but we also have other services like Glacier that we announced a while back as an archival service where you can store a lot of genomic data for a penny a gig a month for future analysis. Also, a lot of pipelines rely on file systems that require policy-compliant file systems. We've been working very closely with other partners, and just this week, Intel announced the availability of the Lustre file system on the marketplace that you guys can just launch now, and you have a policy-compliant file system to run your analysis on. So you have S3 as a data store for your source and results, and during the interim where you're running the analysis, you need a policy-compliant file system. You can just fire up a Lustre file system, and there are other options as well on Amazon Web Services. And then we also have the relational database services that you can use, and also NoSQL data stores. and Redshift as well, where you can, that you can use for data warehousing capabilities. So that's the building blocks, but what excites a lot of people is the availability of these tools that are used in research end up so prevalent on the cloud in the form of AMIs or in the form of platform as a service that a number of our partners are providing. So a lot of the work has already been done. So you're not going to be coming to the cloud and building things from scratch if you don't want to. You can have an entry point. You can build your own cluster. You can build your own pipeline if you wanted to. But there are a lot of tools that are already available there, platforms already available over there for you to get started with. And to top all of it is a very flexible purchasing model. So now you have the compute, you fire it up. A lot of people are familiar with the on-demand compute pricing model where you pay by the hour, but a lot of people are not familiar with is the spot pricing model, which is very applicable in the genomics analysis environments where if you have interruptible workloads who can tolerate interruptions, you can bid your own price and lower the cost dramatically for your compute jobs, and some of which Professor Peter and Dennis are going to talk about how they use spot instances as well. And there are other pricing options also, basically providing you with the flexibility to be able to not only have that kind of scale to be able to run the analysis, but also control the costs because these analyses are going to be huge. You will be working on thousands and tens of thousands of genomes to do any meaningful analysis in this space. So with that, I'm going to hand it over to Professor Peter Tonelato. I think we have to change laptops.
The arrow to the right, remember? Perfect. Thanks. Well, thank you very much for the invitation to come. Dennis used to be at Harvard with us, and he just recently defected to Harvard of the West. So I have to say that now every time we speak. But it's great to see him and to be here to talk to you about how we're using AWS. We actually started using AWS, I think the first time I got on was either 2004 or 2005, and we installed Oracle on an instance, and Oracle got interested in us doing that, and then we started interacting with them and AWS. And we got in a little bit of trouble because they put 1 million simulated patients in an Oracle database on AWS back when there were some privacy concerns. And fortunately, they were simulated patients. So although we got a lot of activity and discussion about that, AWS was happy to hear that they weren't actual patients when we did it. So, and since then, we've had a great relationship, and it's culminated in this work that we're going to talk about today. My part of the presentation is to talk to you about the very direct impact the work that we're doing on optimizing our whole genome analysis on AWS is going to have on actual clinical care, especially in cancer. And our ultimate objective is to participate and work with the Multidisciplinary Breast Cancer Tumor Board, which is the clinic that every week meets to see each patient who has just been staged or it's just been diagnosed for breast cancer, and who decide, there's this group of about 20 physicians who get together and decide exactly how they're going to treat this patient and the progress of that. And what our objective is, and they go around the table, and each group, the radiologist, the pathologist, the oncologist, they all present the evidence that they have about the type of the cancer, the staging of the cancers, the diagnosis, the prognosis, and deliberate on the treatment. And what we're doing is introducing the results of whole genome analysis on their somatic, that is their cancer genome, and introducing that data and information as part of that deliberative process. And so our target is to conduct whole genome analysis in what we call clinical turnaround time. So unlike the Baylor and many other studies, which we are a part of too, we have 6,000 autism genomes, and Dennis will talk more about that a little bit. We do a lot of analysis, batch analysis of whole genomes, but that's really for scientific discovery. What we're doing here is for clinical utility, and it's a very different approach to using AWS. And the hospitals are very sensitive about how we do this. The physicians have a very high standard about the reliability of the information that we extract from those cancer genomes and how that information might be used. And so our objective is very simple. We don't think of this as a computational optimization project, although at the nuts and bolts level it is. What we want to do is, once a whole genome worth of sequencing comes to us, we want to conduct the analysis in less than 3 hours, and we want it to cost less than $100. And everybody knows about Obamacare and the prices of everything and this and that. It's been a lot of discussion, obviously. Well, we're trying to make clinical whole genome sequencing a very affordable, I shouldn't use that word maybe, but an affordable aspect of clinical cancer care. And by the way, we don't have a website, so we can't be blamed for any of that problem. So I'm just going to quickly go through a few, kind of give you the context, and then Dennis is going to go through and tell you what we've actually achieved to date. So to demonstrate that this is not some trivial exercise, I just wanted to put the slide up about all of the individuals who are participating, and these are really the chief of various divisions on the physician side. There's surgery, genetics, oncology, pathology, radiation oncology, imaging, genetic counseling, social work, and then there we are on the far right, the bioinformatics team. And they are all participating in this and trying to understand how this analysis that we're going to talk about today can actually be incorporated into this very complex treatment protocol, that when a patient comes into Beth Israel Deaconess Medical Center and they are being referred there because of possible breast cancer, evidence of possible breast cancer, the process that they and their samples go through in terms of determining what is the ultimate treatment. And our objective is to incorporate our work into this very complex, and not to disrupt the typical way that a patient is treated. And so I don't want to go through all these steps, but I do want to make it clear to you that we can't just sort of sit around and say, well, how do we do this computationally really fast? That's not enough. We have to do it fast. We do have to do it fast, but we have to do it in a manner that will fit in the clinical practice, the best practice approach. So to give you an idea of how frequently we might have to do this as our effort goes forward and as the whole genome analysis becomes part of it, in a 22-week period, we're talking about several hundred cases of breast cancer. So that works out to be 5, 6, 7 cases every week. And so at the current rate of genome analysis and digestion of the results and annotation of it and extracting from that annotated results the clinically actionable information, if you think about the pace of this kind of number of, and this is only breast cancer, so this is not all the others. Just in breast cancer, you're probably having to process several cases every day or at least every weekday. So that 3-hour, $100 turnaround time and then getting it back to the team so they can digest it and act on it is very important. This is just another measure of the types and numbers of cases that come in the Beth Israel Breast Cancer Tumor Board on a very regular basis. It's a very important aspect of how we think about what our technology and even how AWS can play a role in fitting into this system. I put this slide together because whole genome analysis, that's what the WGA stands for, actually plays a role in virtually every aspect of cancer. Conducting data from whole genome analysis can be used to conduct and calculate the risk to cancers, including breast cancer. Is used very commonly in tumor characterization, breast cancer tumor characterization, the diagnosis, the prognosis, prediction of response and outcome to targeted therapies, some of which are very targeted to very specific breast cancers. And then can also provide indications of what kind of outcomes and possible recurrence, even risk to recurrence of cancer. All of this can come from clinical whole genome analysis, and it's quite amazing how that exercise, what we think of as maybe a computational exercise, can really play a role across the entire spectrum of the cancer, the progression and treatment of the cancer. Another aspect that I want to get across to you, although it's not particularly about AWS, but it shows you the impact of the success of our approach. There are currently there's actually about 40 devices now. When I say devices, I mean multiplex genetic test kits, which are being used at various levels in cancer centers for conducting analysis of the tumor, of the genome, or the so-called somatic genome. And then after that kit is exercised, kind of like a blood lab test, but it's a genetic test. After that test is done and the data comes back, that data can be used as part of the treatment plan. Well, whole genome analysis can replace, another thing that's important if we go back to affordability is every one of these tests costs on the order of several $1,000s. And so you can imagine if a breast cancer patient comes in, or maybe someone at risk to breast cancer, and they start getting these different tests, each and every one of them costs several $1,000s. It's very easy to create scenarios, very average scenarios, where 5 or 6 of these tests are performed over the course of a few years as the cancer progresses. They're talking about $20,000, $30,000 of genetic testing as part of the natural course of cancer being characterized and ultimately treated. It's another important aspect of why we want to do whole genome analysis. When you look across all of those tests, there's literally thousands of variants that come from the genome that are being tested for. So if you have to go through each one of those individual tests, again, each one costing several $1,000s, maybe you're getting a few hundred variants, whereas if we can replace it with whole genome testing, perhaps at the same price, only a few $1,000s, including the $100 to do the analysis, all of these variants would then be detected as part of that analysis. So it's very important to us to, again, characterize the use of whole genome testing just like you would do any laboratory test as part of the clinical care. And this workflow sort of gives us an idea of how that might be conducted. A patient's sample comes in, the next generation sequencers that digitize that genome are run on the genome. And then what Dennis is going to talk about, what I'm sort of leading into is okay. Does this just pop up? Let me just get back to... Okay. So again, the bioinformatics analysis that we're going to focus on for the rest of the talk takes place as the third step. And then a very important part, which we're not going to take any time to talk about is how we digest those results and present them to the physicians in that multidisciplinary breast cancer tumor board in a way that they can understand, digest, and use that information. So in order to conduct that analysis, there are actually a number of different analysis pipelines, all of which may be called on and used on any given situation. And we set up each and every one of these. We're going to focus today on the DNA sequencing pipeline, though it's represented by that upper kind of area in this diagram. And all of this emerges from the very rapid decrease that's been demonstrated in the last, especially the last 7 years since the so-called next generation sequencing technologies have been introduced that have brought the cost down some billion-fold in about the last 12 or 13 years. That billion-fold decrease in cost of sequencing has now made it possible for us to suggest and now try to integrate whole genome sequencing and therefore whole genome analysis into the day-to-day operations of a multidisciplinary breast cancer tumor board. So again, I just want to emphasize we're trying to not think of this as simply a technological achievement, but rather as a component within a very natural flow in the clinical setting. Currently, if you think about what's happening in clinical turnaround, the sample collection takes something on the order of 8-10 hours, depending on the course of the patient coming into the hospital and when they see the physician. When a sample is decided to be taken, they walk down to the lab, wait an hour down there, and so on and so forth. The sequencing takes several weeks. It takes quite a bit of time, so we have no control over that. The analysis today for a fully sequenced, very high coverage somatic genome takes on the order of around 40 hours. And it depends on, of course, how much CPU cycles and resources you're willing to throw at that, but we think of it about 40 hours. And then clinical action, usually the results come back the next day. The multidisciplinary tumor board then meets. They meet on Wednesday typically, and then either Wednesday afternoon or the next morning they go and see the patient and make a determination and talk with the patient about the treatment protocol. So we don't have control over, our bioinformatics group has virtually no control over three of those 4 areas. What we have control over is the analysis, and that's what we're going to focus our attention on. So what we want to do is change that from 40 hours and on the order of $1,000 or $1,400, and the optimization that we're going to talk about later is pushing us towards whole genome, again, under 3 hours and under $100. Right now, as I mentioned before, if you take a whole genome and do the analysis, it takes not quite a day, but it costs on the order, well, this is actually a few years ago when we first did it. It took about a day. And again, it depends on how many CPU cycles you're willing to throw at it. But it costs about $1,500 the first time we did a whole genome analysis on AWS. And what we're going to do now is talk about where we're at and how we got there by conducting a series of optimization steps.
Ooh, wow, it's on. Okay. Wake up. Thanks, Peter. I'm Dennis Wall, and I, as Peter indicated, just recently started making the transition from Harvard to Stanford. Definitely not the Harvard of the West, though. All right. Operationalizing the process of clinical annotation and interpretation of genomes is not a trivial problem, and it's something that I think all hospitals have to solve. And Peter already gave a great introduction to that. I mean, there are time sinks that are associated with getting a little feedback from this, with the decision point to move towards full genome sequencing. So you wind up with a tumor that's progressing. You make a decision about that tumor. that might be something like treat with erlotinib, a particular chemotherapy agent, but under that circumstance, unfortunately, the patient doesn't respond like normal, like what you've seen before in the past. And at that point, hospitals are increasingly ordering genome testing for individuals that don't respond to the first line of chemotherapy for a particular kind of cancer. And at that point, of course, they need to understand the somatic mutation landscape. What's going on within the tumor itself? Is it adaptively evolving away from the chemotherapy agent, or has it already adapted randomly away from the chemotherapy agent that's targeting a particular pathway that enables the person to be able to metabolize that chemotherapy? It's just like going back on me. Okay, can you hear me okay? So we've been working really hard to try to operationalize the other part of that time sink, make a decision to begin sequencing.
It's upside down to my optics.
We can create clinical interpretation of whole genomes within a time frame, as Peter indicated, of less than 3 hours, so that decision support can be provided to clinical tumor boards in a time frame that can actually help the patient. The sequencing will take somewhere between 4 and 6 weeks, and that may be repeated over a period of successions if the tumor tends to, if the individual is not responding to the therapeutic agent that's delivered. So I'll get into the technical details of what we've been building as part of a real-time clinical decision support system for Beth Israel Deaconess Medical Center and also for other medical centers, and hopefully increasingly something that we can deploy on a broad scale across not just major universities and academic hospitals, but also community hospitals. I think another important point is that, at this point in time, universities and academic centers and hospitals don't have the capital to spend on significant infrastructure for interpretation of whole genomes, and yet they need them, and increasingly that will be the case, that there is this, there's no doubt that over time, especially for cancer, but also potentially for other areas, many hospitals are going to need to deploy mechanisms to do whole genome sequence interpretation of cancers that are coming through their hospital. And right now that includes primarily 5 major cancer areas: colorectal, renal, lung, breast, melanoma. But the research field is moving rapidly, and a lot of new variants are starting to pass the threshold from research to clinical utility. And as that happens, there's going to be an increasing need for dynamic, affordable, economically scalable systems that enable hospitals like community hospitals in particular to be able to nimbly adapt to those new discoveries and make their hospital state-of-the-art with respect to cancer care. All right, so the system that I'm about to describe is one that we call holistically Cosmos. The idea is to be able to take advantage of EC2 in a number of ways. Our initial foray into this was through primary instance types that are offered through Amazon Web Services and not through Spot Instinct. But over the course of this talk, I'm going to begin to tell you that we've been able to leverage Spot Instance pricing to be able to drive costs down to the area that Peter was alluding to, under $100 for a whole genome sequence. The input types, for those of you who don't know, that go into a data processing pipeline for genomic interpretation are BAM files. These are coming off standard HiSeq machines that are used ubiquitously right now across the United States and elsewhere. These are machines that enable precision sequencing, and they do so in a variety of different, a few different ways. And that field is also dynamically evolving. And like Peter said, we don't have a lot of control over how long it takes for sequencing to happen, and how, and we don't have control over the choice of depth of coverage, for example, that the clinical facility is going to make on what they consider to be clinically acceptable thresholds for depths of coverage. And if you guys don't know what I'm talking about and you want to ask a question, please feel free. These BAM files can exceed, they're very large in size, but sizes that are acceptably large for Amazon Web Services and S3. And we want to be able to take these BAM files, which are aligned pieces of the genome of that individual's tumor, and process them through a pipeline, through alignment process steps, through appropriate checks and balances for quality control and assurances, down to completely annotated with respect to our current knowledge in cancer today. And we're going to be doing this, obviously we want to be able to massively parallelize this across as many different, we want to be able to do so if we're getting 5 to 7 breast cancer patients per week, and we need to do full genome interpretation on those, we need to be, that's just breast cancer, we need to adapt to do all major cancer areas, and we want it, so that number is going to increase from 5 to 7 to somewhere in 20 to 30 for a single hospital. And that, of course, going globally becomes a significant number. And so this is a scaling problem, but also it's a scaling problem that's incredibly amenable to the cloud services provided by Amazon. And so we have done this. We have created a dynamic cluster with instance types that are adapted to the data sets that we need to work with, that are the data types that are coming from Illumina HiSeq machines. And we're putting those into the S3 bucket, and we're launching a well-balanced and highly configured parallelized infrastructure to enable simultaneous interpretation of many different cancer genomes. And we're using currently, and this is increasingly becoming more formalized and industrialized, spot instance pricing that's available on unused capacity that's out there within the Amazon Compute Cloud to be able to keep the costs, or force the cost essentially to stay below that $100 mark, which enables us as a lab to be able to scale up because we're not dealing with oodles of funding to be able to interpret these genomes. But it also will enable the world to be able to take advantage of this kind of compute capacity to do the same sorts of things that we are doing now in our research clusters at Harvard and Stanford. So the optimization procedure that we go through first begins with identifying the correct type and number of EC2 instances, and also configuring and understanding the cluster type. I better just exit that. This is what you get for using your own computer. So currently we're using a non-optimized master, which is, you can read the slide here, it's an extra large, high memory machine with high input-output, and a non-optimized as the master node, and a non-optimized worker, which is currently, and this is some of the stuff that we've been testing over the last several weeks and months, and starting this actually in whole genome data analysis and testing whole genome data analysis earlier this summer, and then really ramping that up and configuring the system over the last several months. So we have created stable clinical whole genome AMIs. And this is, I think, a really important point that speaks to what my previous point, which is that we want to be able to do this in a way that enables us to scale. So we have to have machine images that can be mirrored across the number of machines that we need. And we also want to be able to provide this kind of computing capacity to other groups that are out there and interested in doing clinical decision support with whole genome data analysis. The applications that we utilize that are configured within each AMI are currently what would be considered stable, state-of-the-art tools used in whole genome data analysis and interpretation today. Now, these are cassettes that can be configured as the field moves ahead and advances its ability to do better detection of variants, to do better detection of genomic lesions that are not single nucleotide variants, such as insertions and deletions, and also more sophisticated and faster types of tools that go beyond what we believe to be state-of-the-art today in terms of alignment speed. We also configure the AMIs with the human reference genome to enable comparative analysis and to be able to do the alignment, to be able to provide the variant calling. And we provide annotation databases in the AMIs that are also fairly stable, well-vetted, and trusted resources that many research groups and clinical groups across the country use for proper annotation of a clinical genome. So the compiler, as you can see here, GCC 4.6, has refined GCC parameters that we can get into if you're interested. We have compressed libraries. We have refined Java parameters for GATK optimization. GATK, for those of you who don't know, is a platform provided through the Broad Institute in Cambridge. It's the genome analysis toolkit. Again, it's considered today to be fairly state-of-the-art. It's one of the most widely used resources for whole genome data analysis. The thing about GATK, though, that we wanted to improve through our configurations in Amazon is that it is not out of the box ready to be parallelized and distributed across EC2. And so a lot of the work that we've done and the scripting that we've developed and the code and software that are deployed within these AMIs are designed to be able to manage GATK in ways that are not currently possible out of the box. And so the cluster disks, by the way, that we're using are Gluster, which has so far been an acceptable choice in terms of its capacity in size of 50 terabytes. S3 storage is where we put the BAM files and transfer BAM files and other files. This is the checkpoint after each successful workflow. This is if we're depositing intermediate files, we can deposit them there and we can check for quality control. And this is where we put storage timing information. So all of the work that we've done so far has been carefully benchmarked so that we can report out, and we're going to be publishing a lot of these results in the near future. The benchmark timings that we have on both exome analysis, which is some of the most commonly used kind of sequencing for clinical interpretation today because it's cheaper than a whole genome, but also benchmarking on whole genome analysis, which is significantly larger than the exome itself. So I'm here to say that we're really close to having this complete, that we're really close to having a confident solution for use of spot instance pricing that meets this goal, this 3 hours, $100 goal. DNA sequencing data come to us, and at this point, we're agnostic to the source. I mean, at this point, we're just going to configure this pipeline for the clinical goal of getting something back to the physicians to make decisions about patients. We take the standard procedures, and right now we have somewhat reasonable, I think no aficionado with respect to the configuration of parameters of BWA and GATK would argue with how we've configured these parameters. They're appropriate for the kind of interpretation that we're dealing with and the depth of coverage that's associated with each of the sequences the genomes that are coming through. We're using BWA for alignment, GATK for processing, and Picard for other steps, and we wind up with our SNP calls and our indel calls, and that becomes the fodder for our preclinical and clinical variant annotation database. This database is something that's been dynamically evolving over time. It's constantly refreshed to reflect the changes that occur within individual databases that are pulled into our genome annotation resource. And so we, so far have set it up so that it is up to date and dynamically updated. The preparation alignment phases look like as they are shown here. Again, this is standard best practices using the genome analysis toolkit. We have preparation and alignment phase. We go to variant calling. We go to annotation. This is all happening using EC2 and using spot instance pricing. Preparation alignment historically is taking the largest amount of time. And those of you who know about genome analysis would agree that this tends to be the largest in terms of consumption of compute capacity. This test is on 5 exomes. And I wanted to show you this so you can get a handle on that. We're doing a lot of careful benchmarking to understand where are the biggest bottlenecks in this process and how can we optimize efficiency. Peter mentioned that our goal is not to do this as an exercise in computer science. That's not at all what we're interested in here, but it's a necessity. And so understanding the bottleneck within each of the steps of the workflow is a critical part of this process, and I think it's something that we've been able to do fairly carefully so that we can understand how to configure the system to be able to overcome those bottlenecks. Know where and when to be able to light up new instances, how many instances, and of what type. So the Burrows-Wheeler aligner are to date anyways, although this is changing. The field is constantly evolving towards better aligners. This one right now is one of the best, and it's the one that we use within our AMIs today. This enables us to take the raw reads and map to a reference, mark duplicates, do quality control, and come up with appropriate alignments so that we can do variant calling. All right, so next I'll talk about creating a standardized and robust clinical whole genome analysis workflow. Again, this is a little bit repetitive. Obviously, I'm saying this over and over again, but it's important to keep stressing that our goal is to be able to get through this process in under 3 hours. And right now, most places working with reasonable resources are at 40 or more hours. And so that's unacceptable. We want to be able to get it. So if we get sequences from the sequencing facility that's used as the provider for clinical whole genome analysis at Beth Israel on Tuesday, we want to be able to turn the data back to the clinical tumor board on Wednesday, which is typically when they meet. Genome key is our annotation. This enables us to, genome key represents the set of pipelines and workflows that are associated with the output of our alignment algorithms, the interpretation of the variants, and contains all of the databases that are necessary for both preclinical and biological annotation, as well as clinical, appropriate, and in this case, clinical, really what I mean is can be acted on in real time in the clinic today. So this will be a V600E BRAF mutation that tells a particular tells a physician that the patient cannot metabolize this chemotherapy, but likely can metabolize that chemotherapy. And that particular piece of information is enough to likely save that person's life or prolong that person's life. The databases that are integrated are these. We have Cytobank, Cosmic, Drug Bank. These are common. Many people use these. They themselves are updated individually. Having all sorts of technical problems today. Anyways, you see the databases. There's now well over 35 of them, and these are the ones. Again, they're all informative. They all have their own strengths, and they are largely complementary to each other, and they provide us the ability to do a sophisticated and comprehensive job at annotating the genomes. And with these, we're able to produce reports in that time frame, in the time frame of under 3 hours. For those of you who care, we have support for generic database file formats like BED. A lot of this, I'm going at a very high level, but certainly if you're interested in diving more deeply, please ask questions when the time comes. So we're also moving, as I mentioned, the BWA today is historically, up until to this point, BWA has been the winner in terms of speed and accuracy with respect to alignment, but there are other tools out there that are emerging, and that will happen over and over and over again. And there's no reason for it not to happen. There are a lot of methods that are being developed in many different labs, and we want to be able to take advantage of that. So the AMIs that we've configured can plug and play with different kinds of toolkits like these things. So SNAP is something that we're looking at right now. And we're also looking at re-implementing, like I mentioned, we're able to look at the timing data and see the benchmarking curves, and we can understand where are the slowdowns, and can we configure things differently to stop them, to increase the speed. So now Cosmos is our stabilized workflow for clinical whole genome annotation, a new workflow and distributed computing platform that we are using. A lot of what I just described represents what Cosmos can do. Cosmos is a generic platform for pipelining many things, and it's a workflow management system that enables job splitting, job tracking. It has a web interface. So Genome Key is embedded within and uses Cosmos. And Cosmos is a series of Python scripts, essentially, but more than that, it's much more sophisticated than that. But we use as our primary programming language Python to workflow this process. But it's not necessarily suited just for genome sequence data. It's suited for many other things. Right now, we've configured it for genome sequence data. So talking specifically more about the Cosmos parallelization, we can look at the benchmark and timing data on the exomes that we've done so far. I put these up because these are the most stable benchmark timing that we have. We have a lot of benchmark data on whole genome analysis as well, and we're getting increasingly more data on that. For those of you who don't know, exomes are substantially smaller than whole genomes. They represent roughly 1% of the genome. So, there's a big expansion in size when you go to whole genome. But these benchmark timing data are going to be the curves are going to be statistically indistinguishable from the curves that are associated with the whole genome data. And so we're using that for more cheap testing within EC2. Cosmos enables job splitting, so you can take a significant job like this one and begin to configure it so it knows how to split it and how much resource capacity this particular job is going to need. So this is just an example of a really zoomed out version of a directed acyclic graph that Cosmos builds to be able to configure the machines appropriately. So it uses the concept of DAGs to be able to create the process flow, and then that process flow tells, automatically in silico tells the next process, how many nodes need to be used for this job. So the preparation and alignment processes are massive. They don't fit on the screen, but you can get an idea of just the size of these DAGs are really big, and the width would go across the entire room. There's billions of jobs that are being done simultaneously to be able to do the alignment. And then as you get down through the process into variant calling, you need significantly fewer resources. So the Cosmos pipeline enables us to configure EC2 spot instances to take advantage of what's required for preparation and alignment, take advantage of what's required for variant calling, and then do a slight, secondary expansion for annotation, so we can go across all the different resources that are available to us for both preclinical and clinical annotation. The web interface enables us to look at the jobs as they process in real time. So, here's loading something. BAMS probably, can't read it, but you get an idea. So the green bar here is meant to indicate the level of completion of that particular job. So you can watch, and these will all kind of run simultaneously. Some are dependent on the previous, and so they won't all run simultaneously, but those that can run simultaneously do. And you can watch real time, see how this job is running. This gives you an idea from the clinical perspective. You just light up this web interface from our bioinformatics clinical core, and we can tell we can say to the tumor board, we're going to be ready tomorrow to be able to deliver results. We can watch the processes happen as they're happening. So that's helpful, obviously. I think it's an important part. So 4 approaches to optimize and achieve our clinical turnaround time. Obviously, we're highly leveraged by the availability of Amazon Web Services. Just the need, this is an incredibly, this process is inherently in operation in fits and starts, right? So you need 1 billion jobs to be done at one time, and then significantly less than that to be done at another time. To be able to do that, that's just configured specifically for Amazon. This is exactly what Amazon Web Services provides. That's why we're trying to take advantage of it. Privacy issues, of course, notwithstanding, and we can talk more about those later, but for now, just suspend your disbelief and and assume that these data that we're able to put up on here we have set things up so that they are extremely secure, they're extremely firewalled, they're de-identified, and we could take advantage of the number of nodes needed at any given time. We have been able to refine and improve various whole genome analysis applications. This is after looking at the bottlenecks within the process using benchmark timing data. And we're creating a standardized and robust workflow. This is Cosmos. Cosmos enables us to create something that is clinically robust and therefore can pass the threshold of acceptance of things that are appropriate for clinical interpretation. So this means it's been vetted in a way that machines that go into CLIA, CAP-certified labs have been vetted. The thing that you put into a clinical service has to be vetted in a very similar way. Depth of coverage, quality control assurance steps, the way you do the alignment, that you know for a fact that there's no experimental noise associated with your call, that there is a V600E, BRAF mutation, that it actually is there. It doesn't need to be experimentally validated anymore. And then the fourth is to stabilize, and I already talked about this, through Cosmos. So whole genome analysis, pre and post optimization, just to give you an idea. Before, $27 for one exome, $27. After optimization, $10. 5 exomes, $48. After, $27. 10 exomes, $90. After, $47. So we're, these slides are meant to give you an idea of the potential of scale and savings that are associated with optimization of this procedure. And we're nearly there, and again, we are right at the point where we're confident we have a system for clock time of under 3 hours and cost of less than $100. So this is my last slide, and I think that's pretty much all I want to say, besides thank you. It's a pleasure to be here. It's wonderful to have the invitation, and I think at this point I'm happy to take questions. Yeah.
So I didn't quite understand, when you're trying to parallelize this, you're using Gluster as the essentially shared file system underneath all the instances you were running. And then were you running some type of grid controller on to manage those instances, or just launching them up through Amazon, or using Autoscale, or how were you managing all of those?
So we created our own solution in the lab that we use for management and configuration. It's something that we haven't finished the software design on yet, but in order to be able to use the spot instance pricing options, in order to be able to manage the clusters, we created our own home-baked solution called Rainforest. Yes?
Are you using that MPI IO splitter or the regular splitter?
The regular splitter. Yes?
Is any of this amenable to using elastic MapReduce?
Not the way it's configured now. We've worked in the past with elastic MapReduce and ran into, for these kinds of data problems, issues in dealing with the Hadoop distributed file system and being able to access, configure it. So the Gluster represents a good choice for this because it's the right, it has the right capacity for the kind of data sizes we need, which grow and shrink over time. So there'll be log files that get created. But getting access from HDFS to S3 to do things like pull down reference genomes and became prohibitive, and so we ended up not doing that. So the answer is, I can imagine it's certainly possible, but this is a better solution. Yes. Yes. Sorry? Oh, are we going to publish the validation results that are represented with validating the process? Yes, we're going to be publishing. We're in the process of finishing the paper now. And the paper is intended to show the benchmark and timing data and the validation steps. Yes. Sorry. You. I don't know who was first, so why don't we go with you first? So the question is, why did we choose to deploy this with GATK best practices? In our own experiences, that's been, and this is based on experimental validation, basically of variant calls being confirmed. The best practices are truly, in this case, the best. But I think over time, they'll be better, and we'll start to realize changes. So I think it's important that you keep in mind that these systems that we're trying to configure now are meant to be configurable. And so it is possible for you to go in and make those. You could change different parameters if you needed to, or felt like for whatever reason, you don't believe that parameter should be at that point on the dial. Yeah.
The question was regarding your clinical annotation database or knowledge. How did you build up that knowledge base?
Yeah, so some of the databases in there are from other places, like PharmGKB, for example, or Drug Bank. And they're managed and maintained by others, and they have their own access and restriction policies and so forth. So we make sure we abide by those. But some of the other data that we use for clinical interpretation of breast cancer, for example, or of melanoma or lung, are things that we've done manually in our labs or in collaboration with other labs. And so we're in a position, because we're associated with the medical Beth Israel Deaconess Medical Center, Stanford Medical. We can work with the oncologists and have them help us vet the annotation that we're doing. And so that's what we've done. We've done that for 5 cancer areas to create cassettes of knowledge that are plugged into this source. And they're knowledge cassettes that are specific to those cancer areas. So, but other stuff's pretty generic.
Are you planning to offer these as commercialized like a service? People can give you a bound file and get back your report?
We have considered many different options, and that may be one of them. But at this point, I think we're interested in trying to create. I mentioned earlier, we want to kind of enable. We want to be able to not just operationalize our own practices, but to be able to operationalize others. And so one way to do that is to just bundle up the Emmys and put them out there. And that looks, we're going to be publishing some of this, and that'll be part of the publication. But over time, yeah, I think, there are services, at the risk of sounding like a salesman, Foundation Medicine, for example, is working on this very thing. And it's important, and it's going to continue to be really important, and to go big and to be able to service the community in need, commercialized enterprises like Foundation need to exist. Yeah.
Cosmos available publicly now?
Is it publicly though? I don't know if it's actually like in a GitHub, like publicly accessible. No, the answer is no. Yes, please come talk to us after.
Per patient, you're doing germline and tumor, right? So that's you collected those.
Yeah, great point. I don't think we ever made that point. We do have match-controlled tissue on all patients. Yeah. So then that's the baseline for comparison to understand the somatic, mutation landscape.
Are you using GATK1 or 2, and is that configurable?
It's configurable. I believe we're using GATK2 as of now. Yep.
We adapted various ways.
Yeah. Yes.
Kristen, so it looks like you're trying to shard up the input reads to run BWA pieces across them to make it go faster. There's sort of a trade-off of using spot instances of the time to load up the whole reference genome into memory. What's been your experience? Do they run long enough that it's worthwhile doing that?
Yeah, so far it's been worthwhile doing that, but it's a good question. I mean, it's still a dynamic. We're still tuning things, and it's not right now. It's been worth it, and we've done that.
Can you go back to that workflow timing slide?
I'll get there. God. There we go. Oh. Let's do this one.
So we've digested this in a number of different ways. I don't know who mentioned that, but we're trying to get a more fine resolution of the massive split, which is part of what we're trying to do.
Yeah. So we've done it and it's been doable, but it's time consuming and we may be able to configure it differently.
There's too many moving parts sort of still right now. And we're trying to get a better baseline before we modify too many other things simultaneously.
Other questions? Great. Thank you.